recorded on Thursday, September 18th, 2014. Independent variables. Hey everyone, I'm Dr. Kiki, and today we're going to fill your head with backyard chickens, a star inside a star, and some psilocybin. But first...
Disclaimer, disclaimer, disclaimer. It's time again for a batch of good news. When it comes to our troubles as a species, it usually boils down to being our own fault. Our obsession with cars and gadgets and technology have brought us into an age of acidifying oceans, melting glaciers, bad air quality, and along with it an attitude that can only be described as use it or lose it. So where's that good news, you ask? Science and technology has won one this week. Scientists in Australia, after just 3 years of experimentation, have successfully found a way to print solar panels on flexible plastic or metal hardly thicker than a piece of printer paper. What's more, they can print the equivalent of one panel every 2 seconds. This stuff could end up coating buildings, cars, or just about anything else that can be coated in a thin plastic sheet. It sounds like we are well on our way to renewable, clean energy that is affordable and easy to use. We can pull ourselves out of this mess, get us back to where we need to be, and maybe even get some cool new gadgets along the way. It all came from some experimentation and teamwork. We have the power. Now we just need to band together to use it, support it, and spread the knowledge of it far and wide. And do you know the easiest way to get started with that last bit? It's called This Week in Science, coming up next.
I've got the kind of mind I can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to everyone! Good science to you, Blair. How's it going?
Good science to you.
On this fabulous, fabulous Thursday evening. Welcome, everyone, to a new episode of This Week in Science. Today we've got a great show ahead, full of, yeah, you guessed it, science! That's what we bring. Anyway, I have some news stories about artificial sweeteners causing glucose sensitivity, potentially, the inside of a star, and soft robots.
Nice.
Yeah, what do you have, Blair?
I have peacocks again.
Yeah.
3D printed Femme Fatales and backyard chickens.
That's a good combination. Good combination. For a second, I thought you were going to say that you had 3D printed peacocks, but
No, that would be sweet, though.
it would be sweet. But now we will dig into the news. All right, everybody. This week, scientists at the European Bioinformatics Institute, EMBLEBI, it's a lot of letters, they have successfully reset human pluripotent stem cells to a completely embryonic state. So they have taken human stem cells, pluripotent stem cells, and these are cells that have been taken, say, from the skin. They're fully adult cells that have had the clock wound backwards on them chemically, taken the instructions away that make them an adult, and then created a pluripotent cell that has the potential to be any other cell in the body. The problem with these pluripotent cells, however, is even though they've been wound backwards, they very often still contain a lot of the histone modification, kind of the extra stuff that tells them, that gives a definitive role. So if it once was a skin cell, it has instructions buried in the genome that still kind of, it's hard to make it not a skin cell. But you can. And so what they've been trying to do is wind the clock even further backwards to a point at which the cell has no memory of its past life. And that's what they have finally achieved in human cells. And this is something they've gotten really good at already in mouse cells. Basically every organism that they're working on these, except for human. And now, human! Blank slate cells! This is going to be awesome.
The duct tape of cells. Hooray!
Yeah, hopefully. And the thing here is there are a lot of people who get up in arms about taking cells from a blastocyst that has come from the joining of an egg and a sperm, and some people have real issues with embryonic stem cells. So if we can take adult stem cells and wind them back to the exact same point, we could potentially not have to worry about it. Because at this point, there are some uses for the embryonic stem cells that the adult pluripotent stem cells, they don't match up. The embryonic stem cells are just way better at doing certain things.
Right.
So
well on our way, though. That's pretty exciting.
We are on our way. Yeah, so it's very cool. Biochemically wiping the slate clean. And then really interesting story popped up this week that people have been talking about a bit. Years ago, it became common for people who are going on diets to suggest they stop drinking sweetened soda to get rid of the calorie intake and to drink diet soda. Just don't worry about it. Just drink diet soda. You're not getting the calories, but you still get to have your soda and your flavor. But over the years, we're still seeing obesity pop up. We're seeing problems related to diabetes, and there have been many questions and several studies that implicate artificial sweeteners in the problem somehow. And the question is, how on earth is an artificial sweetener that your body doesn't recognize as sugar? Your body doesn't recognize it. It doesn't absorb it. It just is supposed to pass right on through. How is that actually having an effect and leading to possibly causing obesity and glucose intolerance? How's that happening? Mm-hmm. Well, some Israeli researchers have said that what happens is that it alters gut bacteria. And this new study is fascinating. They took they took mice and they split them into 3 groups. One group got saccharin, one group got sucralose, and the third group got aspartame added to their drinking water. And then there was another control group that just drank water with no sweetener whatsoever. The mice drinking sweetened water, all of them developed glucose intolerance within 11 weeks. And saccharin was the one that had the highest proportion of glucose intolerance. They used antibiotics to get rid of intestinal bugs, and what happened is they found blood sugar levels returned to normal. Then they took the glucose intolerant mice and they had collected fecal samples from them before they antibiotically cleansed them and got their blood sugar back to normal. They got fecal samples and fed them to normal mice. And the normal mice who were fed the fecal samples, which you don't just think poop, think bacteria here. They're feeding them the bacteria from these glucose intolerant mice. These normal mice who ate the fecal samples developed glucose intolerance.
There you go.
Within 6 days.
I would kiss you, but I noticed you were using an artificial sweetener, and I don't want to become glucose intolerant, so I'll pass.
I'll pass, yeah.
Everything else got just right, but you just ruined it with that. So this has been when we talk anything about nutrition, this has been that big wild card.
Super.
That keeps coming up. Is regardless of what just this should react with that, the gut microflora is going to be a game changer and going to be involved on a scale and a scope that we have yet to really comprehend. This is a beautiful study. I love this. And it's also kind of insidious. You know, the thing you take because you're somewhat glucose intolerant, perhaps to begin with, makes you even more so. So then you have to only use this. And by the way, yes, there's been plenty of research that shows that you will gain weight quicker using artificial sweeteners.
Yeah,
I
and
always
people
thought it was.
used. Oh, go ahead.
I always thought it was because they've also done studies where consuming artificial sweeteners make you more hungry.
That's the thing that I
Doesn't
had
satiate the
that people
glucose.
have been saying, that
Yeah.
it makes you continues your sweet tooth, so you're going to keep eating sweet food even though you've stopped drinking the
And you're glucose intolerant on top of it.
yeah,
Which is
and your
incredibly
glucose
crazy.
intolerant.
Yeah,
Brutal.
that's true. Double
Brutal chain
whammy.
of events.
Yeah, it makes me question though whether or not it does actually increase the whether that is really what's going on, that you're eating more, or if it is just this bacterial thing,
Mhm.
or if it's a combination of the two. The last part of the study is they did extend it to humans, not just mice. They looked at 381 people, and they found links correlationally. They found links between artificial sweetener use, symptoms of obesity, and elevated blood sugar, and noted a 20-fold increase in a certain kind of bacteria called Bacteroides fragilis that's linked to gut inflammation.
Hmm.
Yeah, they enrolled 5 men and 2 women who don't use artificial sweeteners in at all in their diet in a 1-week experiment in which they gave them 120 milligrams of saccharin daily, which is the FDA's recommended allowance. And they had their blood sugar levels checked every 5 minutes and had glucose tolerance tests. And in 7 days, 4 out of 7 people developed a poor glycemic profile.
So let me get this straight. Eating chemicals is bad.
It's bad in this
Basically,
case.
if you could have real sugar or you could have fake sugar that's made in a laboratory, you should just eat the real sugar. This is what we've
Blair,
learned.
any chemist will tell you that broken down to its basic composition, everything is chemicals.
Thank
Right,
you.
you know what I mean.
Thank you.
Evolutionary biologists might point out, yes, but our bodies are accustomed to using a specific chemical. You put a different chemical in its place, you may get a different result.
Yeah.
Right?
The final piece of the puzzle here is they fed mice poop from the 7 people that were in this particular study, and the mice
Fun.
developed glucose intolerance again.
There you go.
Yeah, so bacteria are definitely the key in this. They don't understand what the mechanism is yet. They don't know what is happening for these artificial sweeteners to kick up the growth of these intestinal bacteria. Why would it change the flora of our intestines so dramatically? That's what they don't understand. But it's a very, just a beautiful study, fascinating, and what is going on? I want to know, but yeah, now, diet soda, I think that's going to be on my, it's always been on my moderation list, and I think it's going to be even more so.
I refuse to eat anything that's got fake sugar in it.
I think it doesn't
I
taste
don't.
good.
Yeah, it tastes terrible, and
Leaves that weird film in your mouth.
It's too bad we don't have a former TWIS co-host, Greg Yen, here.
Right.
To this day, Greg Yen is addicted to the Diet Coke. I've never seen him without one within arm's reach.
Yeah, there are a lot of people like that, and it's, information like this, it just makes you, it makes you wonder what would happen if they switched it out, or
Well, they can't, because now they're
They can't.
completely glucose intolerant. I don't even know what happens if somebody who's that glucose intolerant tries to ingest sugar, like raw
Well,
sugar.
in the study, they just gave the mice antibiotics, and it kind of kick-started. So it's possible, that's what we don't know, is how, if you've been doing this for many, many years, if then it's harder to get back to the right area. But it sounds like if you fed them antibiotics, you kind of had them start from square one in the gut bacteria region, maybe even gave them a fecal transplant from someone who had good gut bacteria, then they'd probably be fine.
Yeah, and the question is still, we don't know exactly. There's definitely an effect on glycemic profile within 7 days, but we don't know how long it takes or what level, to what level to get full on glucose intolerance, which is going to lead to other incredible problems.
We
So
replaced the diet soda soft drink she normally drinks with rat feces. Will she notice the change?
Oh my god.
No.
Oh my goodness.
Oh boy.
Oh my goodness. All right, Justin, do you have any stories this evening?
Hidden human origin story. This is breaking crazy
Ooh.
wild news
Love
coming
it.
to us from Europe.
Europe?
Europe. So Europe has been mostly known to be made up from two very large groups. They're the Near East farming communities and the sort of more established tens of thousands of years in Europe hunter-gatherers. And these two groups sort of coalesced as Europe became more and more appetizing to do farming in. Groups merged, and almost all Europeans have a mix of these two ancient groups. That was until very recently. They discovered there is a third group. And what's amazing about this third group is there is a sort of ancient Northern European folk. I don't know if they've come up with a name for them yet. I haven't sort of seen it here. But this group has been discovered, interestingly, and to the maximum amount they found is 20% in some Northern Europeans. It seems as though all Europeans have some percentage of this third previously unaccounted for group. And it was discovered because they found a genetic link between Europeans and Native Americans. So the group that crossed the Bering Strait, right, headed into the Americas, also has lineage from this ancient far northern European group. Very interesting. So this is only just really been discovered. They went through and they were looking at ancient bones. Even Utsi, the Iceman, has a little bit of this northern group, and he was in the northern Italy, southern France, Alpi region. He was quite a ways away. And most of the southern Europeans have a higher percentage of the farmer group in them. And more than Northern Europeans tend to have more of that hunter-gatherer ancestry. But there's this third group that really got discovered because they were trying to investigate, I guess initially, the link that they discovered between Europeans and Native Americans, which sort of seemed to leapfrog an entire continent. Now they think they may have found, and I guess they're calling this, even older lineage, they're calling, I guess, the Basal Eurasians. Let's see, they had the Near Eastern farmers and their indifference to the previous one. Okay, and then there was the Basal the Basal Eurasians are the hunter-gatherers. So this is a third group added to the mix, lending more insight into the disparate groups that make up the populations of Europe and partially Native Americans.
Nice.
That's fascinating. It's just so I just love the DNA as we're able to tease more and more out of the DNA, figuring out when things happened and when genetic mutations occurred to give us a trail, and it's just amazing. Hunter-gatherers, my peoples. I need to get on my 23andMe.
What a strange thing to say.
Oh, the
Not
23andMe.
mine.
So I keep getting more and more cousins populating that thing, but they're always disparate, distant cousins.
That's funny.
No, I guess I'm sorry. I got to reiterate. It is the Basal Eurasians. That's what this newly found northern group is being tagged.
The Basal Eurasians. Nice.
I don't know. It's confusing. But the idea though here, the idea though is we think we have such a clear picture of this of sort of the spread, the out of Africa outward tree that you always see on the Discovery or the National Geographic story. The one arrow is going out.
Mm-hmm.
Some of the DNA is finding that like Finns and far northern Europeans actually have some East Asian lineage connections that weren't anticipated. And maybe it's because those East Asian lineages actually date back to an earlier predecessor. So they're still mapping this. But from what's coming out right now, it does look like people continue to be mobile. I think still following my theory that people were continually trying to find a place that didn't have people. That the greatest motivator for the spread of humanity across the globe was an attempt just to get away from people.
Well, that was the reason behind a lot of crazy changes in structure of animals. I mean, think about the first animals to walk onto land. It was to get away from the other animals. That's exactly why they were up there.
Get the heck away. This is one thing about life. Life will find a way to get away from other life, because life finds other life kind of annoying at times.
It can, especially when you're trying to get eaten by other things.
Right?
Or fight for space or food, yeah.
A lot of pressure. It's one thing to move because you want to be closer to work, right? It's another thing when you got to move because your neighbor might eat you. That's a little more. It's a little bit more of a social pressure.
Mm-hmm. Yeah, I would agree with that. Hey, everybody, you are listening to This Week in Science. You know what time it is. I do.
She loves our creatures, bright and small. Biped, millipede, no pet at all. Wanna hear about animals? She's your girl. Except for giant pandas and squirrels.
Okay, Justin, you're going to need to do that every week, exactly like that again. For those people watching, we have a nice pantomime going on during the Blair's Animal Corner theme. All right, let's talk about peacocks again. Feel like peacocks are one of those animals that keep cropping back up in studies, just like pigeons or another one that we talk about a lot. The peacocks, there's this constant debate about why is that tail there? Right? So the peacock is one of the classic examples of sexual selection where an animal takes on a lot of physical and fitness hardship in order to be selected for by a female, right? So this big tail, it's clumsy, it's harder for them to fly, it's harder for them to evade predators, it's hard to grow, but in the end, they're more likely to have babies. So it's a winning scenario. So that's kind of the classic sexual selection idea. A recent study from the University of Leeds tells us that it might not be quite the fitness disadvantage that we thought.
Well, what was the study before that we found it wasn't length, it was more girth of the tail? Was that the...
That's exactly what it was.
They were looking at the feet and the sort of general spread, not the height.
They were looking, they put trackers on the females' heads to watch where their eyes were looking, and they were looking all along the bottom. They didn't really stray up above the head of the peacock at all. They weren't looking at the top of the fan. They were looking all along the bottom. So what they found now is that this tail, this huge tail that can get over 1.5 meters long, and that the feathers altogether of this tail can weigh over 300 grams. So do your metric conversion, but that's a sizable weight. So they found that it actually doesn't do that much to their ability to fly. So this professor in charge of this study filmed the takeoffs of birds with full plumage. He did a 3D footage. He then filmed the same birds when he cut off their trains. And he looked at how fast, how easy, the energy output, all this kind of stuff, to see what the difference between the two was. And he found no significant difference. He was looking at the bird's center of mass, their wing motions, the movement of the train, and he, like I said, he calculated the amount of power used to accelerate and gain height. And yeah, it was pretty much the same. So, yeah. it's so interesting because it kind of makes us relook our entire idea behind sexual selection, because the peacock is
Right.
very much our poster child for that.
Yeah.
And he did measure the drag of the train all by itself, and the drag doubled with the train on there, but overcoming the drag was not a significant amount of the power used. It was insignificant, essentially. And so it was a negligible change in the energy required.
So
That
it
being
wouldn't
said,
be a survival cost necessarily.
it wouldn't be, except for a couple of things that you'd still have to keep in hold into account that he didn't measure. So the first is that it could affect their ability to run.
Mm-hmm.
It could
Dragging on the
affect,
ground.
yeah, it could affect
Right.
their flight stability. The thing that he didn't mention that I thought about is it just increases your likelihood of getting grabbed by a predator. These guys' number one predators are leopards and tigers, and if you have 1.5 meters of tail, it's much easier to grab that guy and pull him back down as he's flying away. The other thing that he mentioned is that about 3% of their daily metabolic energy goes solely to making those big feathers in their train, which is a good amount of energy, 3% every day. That's not a huge amount, but it's enough to consider as a cost. So it's not saying that it's without cost, but it's not quite the disadvantage that we thought it was before.
I think it's just it is interesting now that people are looking into it just a little bit
Yeah.
more specifically, that, like you said, this is a poster child, and all of a sudden new questions are being opened up. And so what does it mean, not just for the peacock, but for questions of sexual selection and traits generally? Yeah.
Yeah, exactly. So it's definitely something that when we go ahead and assume that something has a fitness cost, we need to test it now.
So,
Oh.
but here's, and I mean, we're talking about a wild animal, of course, but I'm wondering if there was any sort of human intervention with peacock populations in terms of in terms of the appearance. Sort of like, with dogs. You figure the peacock was probably bred to an extent by royalty in India or something over thousands of years as a royal bird, as something that should be on the ground. I'm wondering, and I'm trying to go, my Google thing isn't working here. I'm trying to pose, is it possible that there's some human influx into the current look of the peacock? Or is this a purely natural evolution of this bird?
My understanding is when these birds were first documented, I guess you could say, that they were in the wild and that they had this look. And that however many, I'm sure it was only hundreds of years ago, but however many hundreds of years ago it was, they were seen in the wild and they were seen with these traits, and that's kind of where this whole idea of sexual selection came from. So my understanding is, as far as we know, they've had these large trains for much longer than human influence could have changed it, but I don't know that for a fact.
Because I mean, we've had they've been, it's been thousands of years of human interaction, sort of gathering these up as symbols of royalty and the feathers being used and perhaps being raised.
Mm-hmm.
I'm just kind of curious, like, not to say that these traits weren't there, but I wonder if how flexible the genome is. How flexible, like within dogs, we're talking about a few hundred years, right?
It's a good question.
Every dog species that we have is a product of breeding at this point. Most of it occurring in the last 150 years or so, 200 years or so. Birds also have a sort of plastic aspect, sort of plasticity to their genome that allows them to make alterations quickly to adapt. And I wonder how even you call it small scale, but I'm talking about a big chunk of India, right? India, small scale, all of India, sort of breeding these birds. Because you figure, that large plumage has got to make them an easier target in a completely natural environment. it seems almost unsustainable to some degree to have done that for a very long time.
But there are examples of other birds that have crazy plumage, things that seem unsustainable or unrealistic to have evolved all completely on their own, yet they're in the middle of the Amazonian jungle, away from human impact whatsoever, and so it's not necessarily so questionable to me that they would have evolved this potentially, but I get where your question is coming from, because the symbolic significance culturally to the Hindu religion, culture, it goes back thousands of years, and so we're looking at something that people have been watching these birds for not just a short period of time, but they've been aware of them for thousands of years.
Is it sultans in ancient India? Am I using that right? I might be using that wrong. Any person of prominent wealth and power in ancient India having a large cadre of peacocks.
Right. I'm fairly certain, though, that they weren't matching up peacocks themselves. Do you understand what I'm saying? Even if they have their own peacocks on their own
property
land, the peacocks are still selecting who they want to be with, or the peahens, I should say, right? So my understanding is they didn't put a peahen and a peacock into a cage and say, all right, you guys go do your business because we've matched you two up. It's also much harder to control that with birds than it is with mammals.
Mm.
Yeah, but I could see,
it's happened with passenger pigeons and others.
Yeah, I can see that being a big deal in the ancient sultan world to have a bigger peacock than the sultan on the other side of the mountain. It's that sort of like you would want not just to have more, but to have them be more royal looking. I don't know. It's a question I'm
just saying you have to ask. You have to ask what the mechanism for the selection for these humans is, because chances are if they think this highly of the peacock, they're not going to kill peacocks that don't have large enough trains. They're just going to keep the ones that have the large trains they want, and they're going to let the other ones loose. So it's not really going to change the gene bank that they have. So I'm not convinced that is an influential factor, but it could be. Moving on. More bugs. You guys heard of emerald ash borers? They're really pretty.
I have not.
But they are also invasive. They're from China, and the U.S. Department of Agricultural Forest Service claims that these borers are the most destructive forest pest ever seen in North America.
Whoa.
Yes. So they have spread throughout 24 states and 2 Canadian provinces. They've killed tens of millions of otherwise healthy native ash trees, these ash borers. And so what they've done at Penn State is they wanted to do an experiment to see how they could control this population. So the way they did that is they found a few different ways to try to trap these bugs, specifically the males, and either trap them and keep them or exterminate them. And so they did it a few different ways. One, when they collected dead female beetles, they coated them with a vapor of nickel, used nickelized shell to then make matching molds in the shape of a resting beetle. Then, by pressing a structurally colored plastic sheet between the two molds while applying heat, they cast replicas or decoys. And then they tried to retain the surface texture of the beetle at a nano scale. And then they painted them metallic green. So those were those decoys. Then they also made decoys using 3D printing. And in that method, they molded the plastic into the size and shape of the emerald ash borers, but did not try to duplicate the texture. It was too hard in the 3D printing arena, the way that they wanted to do it. And then they took just normal dead female emerald ash borers. And they put all these guys onto leaves in the forest to see which of them best attracted the males. And then they also had some of these decoys with a 4,000 volt charge to electrocute and trap the males. when they landed on the females. Yeah. It's a smart idea.
It is.
I think it's a great idea.
Oh, we gotcha. Okay.
They found that all three kinds of decoys, the real dead females and the 3D printed and the nickel cast decoys, all three of them, they elicited flights by males towards them. But they almost always chose to land on the dead females. After that, the more realistic bioreplicated decoys that had the nano-level textural detail. And when they flew towards the 3D printed decoys, they pretty much never landed on them. The males usually would quickly leave the bioreplicated decoys after they landed on them, but the amount of contact that they had with the bioreplicated decoys was enough to give them the shock and stun them and capture them. So then they tried to replicate the light scattering properties of the beetle shell with white lasers, and they painted them emerald as well. And this improved their effectiveness as well. So basically what they learned was that not only is it color and shape of the female, but it's also fine scale texture. So the bumps and spines, all those things are a big cue to those males. And it also changes the way light's scattered when it hits the female beetle.
Yeah.
So if we're trying to figure out how to eradicate pest animals, this is really great information to have, that if we can replicate females down to the fine texture of the surface of their body, we might be able to pull in these males and electrocute them. Hooray!
Whoa.
That sounds like so much work, though. I mean,
Up to the
little
point you said
decoys, have a
electrocute,
setup to electrocute them. I mean, wow. It seems like a lot of work, and I wonder just how effective. Once they get it right, how many traps do they have to set up? How many females do they have to? How many femme fatales do they have to put out there? And how effective will it actually be? Is this the most effective route to go down?
It's a good question. I mean, if you eradicate all the males, you can't really have more females, can you?
Yep.
So that's definitely a way to, if you can take out most of the males, you can stop the species at the generation level. So it's definitely, without having to eliminate every single beetle, you can eliminate reproduction of those beetles. The question is it going to be just slightly ineffective enough that maybe a couple of beetles figure it out and then they have babies that know how to tell the difference between the decoys and the real ones?
Right. Yeah.
But I also, I wonder if there is a slightly better way to go about doing this. But it's pretty cool. It's a cool application of a lot of different types of science.
Yeah, and I think the thing that is most interesting about it is that it's very species specific. You're not going to be drawing in any other species of insects. You're only getting the males of that species, as opposed to, say, some of the stuff that goes out into the environment to target male mosquitoes.
Well, until you discover that birds have been trying to digest these things.
Yeah, there are a lot of interesting questions that this brings up, but I think it's fascinating.
Yeah, it's definitely an avenue to look at. But I also, that was one of the things I loved about this story, was that it was a coordinated effort between a bunch of different types of scientists. Someone had to be good at printing these models, and someone had to be good at pressing these models out of nickel, and someone had to be good at charging these models, and someone had to know the natural history of these bugs. There were so many different categories at play to make this work. I love it.
Yeah. Well, why don't we just chop down all the trees?
Yeah.
I mean, we need furniture and houses, right? Ash, great wood.
Yeah. Well, then we don't need to get rid of the bugs because they were just killing the trees. So
right. They don't have habitat. They won't spread anymore.
We won't give them the satisfaction.
Is it possible we could scale this up to, like, humans?
Now, Justin.
properly textured.
I think that would be entrapment.
And I think that it's used in makeup. We've got nanomaterials and other things being used to get the exact shade of lipstick or eyeshadow that will properly entrap a male.
Touche. There you go.
Yeah, there you go. It's already working out.
Yeah. Do you want to hear a quick thing about backyard chickens?
Yes.
Great. Beware. I know that backyard chickens are on the rise. Lots of people like to have them. Why wouldn't you? You want to make your own food. Why would I buy eggs at the store if I could keep my own adorable clucking chickens and they would give me my breakfast every morning? Well, apparently there are a lot of medications that chickens sometimes need, and a lot of veterinary pharmacological stuff you can get over the counter. And you might not connect the dots, but anything you feed your chicken could end up in those eggs. And so if those chickens are on any sort of medication, a lot of the time the medication will end up in those eggs and it can make people very sick. So you have to look at a few things. First of all, if you have backyard chickens and you're consuming their eggs, you need to always get medications that are specifically for egg producing chickens. They have different medications. They also usually have very specific directions for how long until you can eat their eggs after you give them the medications. There's also a lot of things that they could come in contact with that you might not even realize, so that also can end up in the eggs. So that might be something as simple as something in your water that you don't realize is in your water could get biomagnified in the chicken, like hormones potentially could end up biomagnified in the chicken, and then it could end up in the eggs. So definitely read a lot of labels. Be very careful what you feed your chickens.
Don't give your chickens your antidepressants.
That's also true. But another good example is if you have wasps in your backyard and you spray pesticides for your wasps. Those wasps die and the chickens eat those dead wasps. Where do those pesticides go? They're in your eggs.
I had to give the antidepressants to the chicken, Kiki.
Yeah.
Yeah, lately he's been down on his cluck.
Pump.
Sorry.
So pesticides, weed killers, any sort of pharmacological stuff, beware if you're eating your chicken's eggs.
Beware of eating the chicken as well. But I guess
where's my home analytical kit that can be on the countertop in my kitchen that I can just put a little like thermometer thing into something and it can tell me what's wrong with the food, why I should eat it, why I shouldn't eat it, with the nutritional where is that? Aren't we there yet?
It's the tricorder. We're so close. We're so close with the tricorder.
I think the thing to remember, though, is that if you are backyard chickening or you're getting eggs from backyard chickens, you're still, these are things to consider because, yeah, what you are, what you eat. Your chicken is what it eats. And that is the same situation for farm-raised animals. And those animals are getting a lot more antibiotics and a lot more medications. It's just part of their feed very often. So you very likely are getting a healthier egg by...
If you're a vegetarian?
It's good. It's good to look at that, for sure.
So it's good to figure out what is going into your chicken. You have a handle on what your chicken is going to be eating.
Definitely.
When you're buying a carton of eggs, you don't know what those chickens have eaten or what their life was like. So.
But now that you tell me this, I feel like that might be a more controlled environment.
It depends. It depends on the eggs that you buy. So definitely if you don't have a backyard chicken and you're buying eggs at the grocery store, look at the label. If the egg just says grade A eggs, the chances are there's something else in there. But if you look and it says free range, vegetarian fed, no hormones, then you know you're probably getting a clean egg.
Yeah, vegetarian fed. They're eating people.
Yeah, they're eating waste food. They feed chickens waste food, which could be meat, could be chickens.
Vegetarian chickens. Anyway. Oh, backyard chickens. I think that's going to be the name of my next indie rock band.
That could go on the list.
I think it's going to go on the list. Backyard chickens. We're scraping it up. It is time for us to take a break, everybody. Please stay tuned for more This Week in Science. We have lots more coming up in the second half. Psilocybin, what? I've also got hummingbirds. We'll be back in just a few moments.
You can't believe what a skeptic I am. I can't believe you believe in that man. We disagree, but I still give a damn. Your guru assures if you follow his regimen, you will become a most excellent specimen. The power to live on and on for all days is right at your fingertips if someone pays.
This Week in Science is made possible by viewers like you, by listeners like you. You help us do everything that we do, so thank you. And now I'm going to ask or tell you ways that you can continue to support us. And here's what we got going on. TWIS. We have merchandise that you might enjoy. T-shirts, hats, sweatshirts, tote bags, all sorts of things over at our Zazzle store. So if you go to twis.org, our website, and look for the Zazzle store in the main header, you can find all sorts of cool things. Are you cold? Do you need a sweatshirt? Why not wear a TWIS sweatshirt? That could be pretty cool. Does your friend need a sweatshirt? Maybe they deserve a TWIS sweatshirt. Head over to twis.org, find our Zazzle store, find all sorts of swag that will fit your needs. Also, we are supported by listeners like you through donations, and we have a couple of ways that make that possible. We have PayPal donation buttons all over our website at twis.org, so if you head over there and look at our main page, look at the show page, listen to the most recent episode, maybe scroll through show notes, leave a comment, all that kind of stuff, there's PayPal buttons. Boop, 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 boop. You can figure out how you want to donate using PayPal. It's very straightforward and easy. But for those of you who would like a new and different way to support us, we have joined up with Patreon, patreon.com. Patreon. It's like Kickstarter, where you support us and then we give you a gift in return. So patreon.com/thisweekinscience is where you can find us over there on Patreon. And it's awesome. You support us, you get gifts. It's great. It allows us to get amazing stuff. Like I just bought a new DSLR camera for us to make more TWIS shorts. I'm very excited about this. The video quality, the audio quality is going to go up. We're going to make better productions, and you're going to like things better. We're going to do more stuff, and it's all because of you supporting us on Patreon. So thank you so much for that. Additionally, whatever your preference for donating, just head over to the website. There are links there, and just thank you for that. If you can't afford the donations, don't worry about it. We understand. Everyone's been cash-strapped at one time or another, and we can use your help in other ways. So if you want to get the word out about TWIS, tell your friends, tell your family, use your social networks like Twitter, Facebook, all these other places, share us. Share us online, share us in the real world, hand your friend your media player and say, I think you should listen to this. It's TWIS. People are going to love that. Use your social networks for the good of science and TWIS, please. We'd really appreciate it. We thank you for your support. We could not do this without you. And we are back with more This Week in Science. We have so many stories still ahead. You want a story about the Hummer sweet tooth?
Obviously.
Yeah.
How could you even ask?
The hummingbird. The humming hummer. They don't really hum. It's the sound of their wings. It's wonderful. But hummingbirds, they have a sweet tooth. And you might not be surprised if you've ever fed a hummingbird, right? You get a nice red little feeder and you fill it with sugar water, or you get a clear feeder and you put red food dye in it and sugar water. Sugar water. They come and they lap it up and they love it. Well, it turns out hummingbirds are different from other birds, and chickens, being other birds, I'm just playing this off of your backyard chicken story from before the break, Blair. Chickens do not have a sweet tooth.
They don't care.
They don't care. They're more likely to pick grains than they are sugary sweets. They could care less. They do not even really detect sugar. And what happened is that dinosaurs lost the mutation that allowed them to be able to detect sweet. And that's why chickens don't detect sweet. But what about hummingbirds? How come hummingbirds can?
Yeah,
Researchers...
they're either really old, right?
Uh-huh.
Or they got it back.
It is number 2. They got it back.
That's exciting.
Hummingbirds re-evolved the mutation to detect sweetness. And so there's the sweet taste is recepted by receptors, a family of genes that code for receptors called the T1Rs. The pairing of T1R1 and T1R3 detects amino acids and allows us to taste savory umami, and T1R2 and T1R3 detects sugars. A postgraduate student at Harvard University looked at the genomes of 10 species of birds, found that insectivorous and grain-eating birds possess the gene pair, the T1R1 and T1R3 pairing, that allows them to get that amino acid umami flavor, but they don't have the T1R2. The modern birds evolved from carnivorous meat-eating dinosaurs, theropod dinosaurs. So that's why the amino acid flavor or taste would stick around so much. And they didn't need to detect sweetness. That's why cats don't detect sweet either.
They don't act very sweet either. That's why they're so cranky all the time.
Hummingbirds, however, their diet comes from nectar of flowers, sugary nectar sources, and so they really aren't going for the grains, the insects, that kind of carnivorous or grain-eating diet. And so they have gotten back these receptors. They have mutations in the T1R1 and T1R3 genes and switched them from savory to sugar.
Mm-hmm.
So they re-evolved it.
That's so
because
great.
They needed it. Yeah. The food that was available, the hummingbirds evolved to eat that, and as they evolved to eat it, their taste for it evolved as well.
Right. Because being able to taste the sugary food would make them probably eat more of it, right? Mm-hmm. Which would make them more fit.
Yes. So what the assumption that they're making is that ancestral hummingbirds lacked the sweet receptor, but maybe went to catch insects on flowers. Occasionally, they got some nectar in there. It wouldn't have made a difference at all at first, but then some of them would have had mutations in their taste receptor genes that would have allowed them to taste the sugary liquid. Those would have been able to get would know that it was sweet and would maybe like it more and go for the sweet flavor more often, which is a very high energy source of food. Mm-hmm. It would possibly have given them evolutionarily an upper hand and also allowed them to get a niche separate from other insect eaters. So they'd actually be able to get a very specific food source and not have to compete with the insect eaters as much.
I would postulate it slightly differently. I would guesstimate that they discovered the high energy value of the nectar first. And over time, just to identify the good nectar from something that wasn't producing, the sweetness was that thing that made it different. And that's why that got increasingly more relevant to the population. More natural selection, maybe it's epigenetical, don't know. But I bet you that that's how I would think it would happen. There's
Yeah.
more of an ability to identify which plants they're getting the good from.
Yeah, I mean, it's a question.
Got a pop quiz for you. I got a question. Pop quiz. Pop quiz.
Pop quiz.
Another animal-related story here. Name 3 species on planet Earth that kill their own kind.
Humans.
Parakeets.
Wait, which? That kill their own kind. What did you say?
Parakeets.
Parakeets go around killing other parakeets? Do they really?
Female parakeets go around killing other baby parakeets. They kill. Female parakeets are pretty vicious, actually.
That's pretty.
Justin, I think you need to change your question because I think most animals will kill members of their own species.
Yeah, lions will, humans.
in competition with other groups. Not just, kill the old
As
one
a
for dominant.
group, like they'll
As
gang
a
up
group.
on another member of their species.
Okay. Name
Chimpanzees.
3 species that have organized killing of their own species and other groups.
That's a different question.
Chimpanzees.
Yeah,
Human.
Mm-hmm.
And is there
Well, you've
another
actually answered the
primate?
question. Chimpanzees and humans. That's actually it. There isn't a third. There isn't a third that they've really recognized this sort of behavior taking place in. So this is an interesting. It's a multi-international study of different groups of chimpanzees in Africa. It's a conclusion of a 5 decades long study, but it's really a bunch of studies that have been strung together to paint the entire picture. And the early conclusion had been that chimpanzee group killing of other chimpanzees over territory was actually due to mankind's intrusion into those territories, creating more disputes over the limited resources, and that was increasing the violence and actually maybe even causing it. But this is actually 18 different chimpanzee communities, and what they discovered was exactly the opposite. In fact, the areas with the least amount of aggression between rival chimpanzee groups was happening in territories that were being encroached upon. And the areas with the most violence and the most killing, and the most organized warfare was in areas where mankind was not even present. So this is kind of an interesting thing, this is our closest relative, and it is the most violent, warlike of the species out there.
So congratulations, we didn't invent it.
We didn't invent it. It's not our fault. Inherited trait.
Awesome.
What we're doing. It's just violence is just it's ape in essence. It's just in us, right? So this is kind of funny. I love this quote here. Okay, well, there's considerable variation in rates of killing by chimpanzees living in different populations. So even chimpanzees killing each other is not inevitable. And of course, says the researcher. And of course, we humans are not chimpanzees. Oh, see, I thought we were. We have the ability to shape and alter our behavior in ways they can't. We can alleviate considerable human suffering by harnessing that ability. Yes, if it only hadn't been going on for all of human history, I might agree with you completely.
I wonder, though.
Go ahead.
Oh, wow.
You can-
Buy me a Coke. Not a Diet Coke, though, either. Give me the sugar.
I'm just wondering, though. It's been postulated that the more peaceful bonobos are actually more closely related to humans than the chimpanzee, and if that is the case, then what's going on? That's kind of interesting.
That's
Right.
exactly what I was going to say, is we have the opportunity to take things the chimp route or the bonobo route.
The
Which way
bonobo
should
route
we go?
is love and have sex at least 4 times a day. This is how the bonobos go about their daily routine.
But it's also groom each other, share your food, lots of nice stuff.
And a lot of sex. They have a lot of sex. This is the thing. This is why bonobo doesn't
They're
show
like
up
primate
on the Kids
socialists.
Discovery Channel. It's because they're almost always going at it, right? But that might be seriously, that might be the thing that separates, right? That separates chimp from man is that we went more the bonobo route. We've got a little bit more bonobo in us. We're a little bit more willing to swing than a regular chimpanzee.
And there have been, I'm trying to remember what which researcher it was, but there was a researcher who suggested that in areas where there are large populations of young human males who are partnerless, that violence tends to increase. And that, you know, that when human males are young, they're high at lots of testosterone. Testosterone promotes aggression, and that if males are not partnered with females, or if they're not partnered, they're more likely to become aggressive and warlike.
Well, this would make China like the most violent country in the world. I don't know if there's data to support that.
Yeah, it's a postulate. Yeah, it's an idea. It's an idea that someone's put out there.
Yeah.
Good to think about. Bonobos. We go the route of the bonobos, or more like it. Yeah. Let's share things.
Make love, not war.
Be like a bonobo, not a chimp.
Yeah.
Yeah. What was another cool story I had? Soft robots. Soft robots are going to freaking dominate the world, people.
So I just pictured a teddy bear walking down the road.
So, Harvard University, these people, what is coming out of Harvard? Robot developers from the Harvard School for Engineering and Applied Sciences and the Weiss Institute for Biologically Inspired Engineering have created an untethered soft robot. So it's not tethered to an electrical cable or a controlling cable. It's kind of off on its own. It's a quadruped. It can stand up and run or walk away from the engineers who made it. This robot is capable of being run over. It can walk through snow. It can walk through fire.
This poor robot.
And come out unscathed.
Robots don't care. Fire.
Robots don't care about fire. This is good. This soft robot is going to be the new honey badger. You're right. You're right. Robot don't care. This soft robot, they're hoping, is going to be a rescue tool. That after disasters, it'll be able to get through areas that people might not be able to make it into, and be able to help people out. I think, though, that they will end up ruling the world. I mean, if it can get run over or walk through snow and fire, this robot is, this robot's going to take over the world. It can move in a very scary way. It's segmented. Blair's put a video on allowing us to look at it, the segmented kind of snake-like action. It's got 4 legs. A central motor and processing unit in the central kind of abdominal region. Is that what you call it on a robot like that? It kind of moves itself forward like somebody who's, I don't know, been badly injured in a violent movie.
Yeah, they're crawling to safety.
It looks
they
like
look kind of like
run over by a car.
It looks like if you took Gumby and had him do that crawl that the soldiers do when they're going under the barbed wire.
There we go.
I was going to say it looked like a Jim Henson Muppet. Remember the little tubes that had faces that danced around? It looked kind of like that.
Yeah. It can be submerged in water.
That's creepy.
Yeah. So one of the things about robots to date is that, the world robot domination aspect of it is that, most robots, you put them in water and they're fried. It doesn't work. They submerged this one in water. It was fine.
No problem.
No problem. Yeah. It's a little bit more than half a meter long, capable of carrying about 7.5 pounds on its back, which isn't a lot, but it could be enough for a little rescue kit. It can get into kind of small areas in the way that it crawls because it's not very tall. It's kind of it's got a low profile to the ground. The researchers say as soon as you start thinking about putting the basic components you need to make this work, microcompressors, controllers, and batteries on an untethered robot, you need a design that can carry all the parts. You need to think about something that can handle much higher pressures, so there are material challenges There are design challenges and there are control challenges. The increased air pressure inside is as high as 16 pounds per square inch, which is higher than earlier robot designs. They're using a composite silicone rubber. And yeah, it is one durable little robot. The next hurdle is that they're going to try and increase its speed and also outfit it with sensors so it can sense more about its environment. Because if it's untethered, you potentially want it to be like a little disaster Roomba that can crawl under and around things and get over obstacles.
Yes, Ed from Connecticut. Anybody who will understand the reference, it is like an indestructible Mr. Bill.
I'm just thinking about now, we had that string of stories a couple years ago about different body parts that Japan was trying to get robotic, and I'm thinking about, I mean, obviously you could go the silly route, but I'm thinking more the touch sensors that they had that were basically like fingers. And they could get so much information from these touch sensors that they made in Japan. And if you could put those on these soft robots, that would be a huge advantage, because then, yeah, you could know exactly what you were stepping on, feel your way. Yeah, be so crazy.
Yep. So I think it's a really neat development in robotics to have this kind of a soft-bodied robot that they've come up with.
Oh yeah, panoramic in the chat room there. Yeah, maybe they could take over deep ocean research.
They'd be like sea stars, sea star robots,
cruising around down there, checking things out. So we talked about this a little bit. Davis, California, the town in which I am broadcasting from the moon, outlawed plastic bags.
Yay!
Totally annoying. Totally for it. Great. Totally annoying because I always forget. I've got the totes. They're in the car. And then I go in there and I'm like, I didn't bring them with me. So then I got to take all the groceries out. They take the cart out to the car. Then I'm bagging. I'm bagging in the car. Put them in there. Then they go into the house, and hopefully I remember to put them into the car. Otherwise, I just got to throw everything into the back of the car. Then come home. Then grab the totes. Then go to the car. Then put it in. But here's the thing. Makes sense. Whatever. This is just the city of Davis. I can go to the next town and have them triple bag things. I really need some bags. However, last month, California lawmakers passed a statewide ban on plastic bags, single-use plastic bags that you would normally get at the grocery store and the like. The governor is expected to sign off on this, so this may be the future in California.
Love it.
Yeah.
Currently, nationwide, the number of bags being used, somebody want to postulate a guess how many bags get used, plastic bags get used nationwide in the United
Billions.
States each year?
Billions. It's got to be.
How many billions?
I don't know.
Billion?
Lots of billions.
Hundreds of billions. About 100 billion, actually.
And they're all going into landfills.
Used every year in the United States. Environmentalists have been saying, hey, there's no good way to actually get rid of this at the end. It's a landfill problem. It doesn't break down. It floats away. Birds eat them. Gets stuck in stuff. It's trashed, and it's trouble.
Yeah, it either ends up in the landfill or it ends up in the ocean. Those are the two choices.
Although when I have to say, when they were banning it in Davis, one of the things that came out in the literature is they showed a seagull with a bag over its head. You know, it's why we need to get rid of it. I'm like, when's the last time we've seen a seagull in Davis, first of all? And if so, it's probably already sick. It's probably injured. It probably needed the bag. Something's wrong with the seagull if it's in Davis. But anyway, that was beside the point. So yeah, it looks like California is going to be going bag-free.
I hope so.
The industry spokesman for plastic bag industry, I guess, has a spokesman.
Well, they probably got a lobby too. Right,
and lobbyists. They've been fighting, of course. Warns that the anti-bag campaign could be just the beginning of a much larger war against plastics in general.
Good.
Well, and that's not is that a bad thing?
I guess it just sort of depends on your feel of plastics. I don't know.
There's a couple things with this. One being, there's still going to be plenty of plastic bags needed in the world because of things called dogs.
Right.
Which I hear one right now.
My
What's
neighbor's
interesting,
dog.
though, is I wonder, the environmentalists in general have a way of shooting themselves in the foot whenever they attempt to take one step forward. They take a second step back. They fought against nuclear, so instead we have coal-burning power plants all over, and we have global warming. If we
We've
got
had the
nuclear...
plastic bag ban here for 2 years. I think it's gone really well.
No. I agree. As long as they're also banning paper bags. Because if you're not banning paper bags, then you have a deforestation issue because you've got to double or triple. Now you need 100 billion paper bags.
Most paper bags are made from recycled paper.
Okay.
Because it's brown paper, because it's paper that can be very thick. For all those reasons, it's usually recycled.
And if there's enough supply to replace the 100 billion plastic bags, then fine. But it's just a word of caution. We need to make sure that we're either also banning paper bags or making sure that it's limited to recycled materials, otherwise it won't be.
Yeah, in San Francisco, what they did is they
tapping the recycled market. It's a chain of events. You tap the recycled paper market, so then, we ran out of paper for cardboard boxes. We ran out of paper for this and the recycled. We're using it all for bags. Now we got to go chop down some trees to make cardboard boxes again. Or whatever it is, there's a cycle of life to this.
And then the paper, you take your paper bag and you put it in the recycling or in the compost, or you take it to the store with you again, because paper bags seem to be a bit sturdier than the plastic bags anyway.
I end up just buying a tote every time I'm at the store now. That's my solution. Pay $1. I won't pay the 10 cents for the plastic bag because I'm like, why am I doing this if this is the whole point? But I will.
Have you gotten a TWIS tote yet? You should get a TWIS tote.
I don't have a TWIS anything. I had a TWIS mug. I don't know what happened to it. Somebody stole it.
Oh my goodness. How about some psilocybin to help you quit smoking? Yeah. Justin, this one is for you.
Oh.
Researchers gave people who are trying to quit smoking psilocybin pills. And they found that those people who had more of a hallucinogenic kind of mind-altering effect from the psilocybin pills were more likely to actually quit smoking. We don't know exactly why this works, but psilocybin pills for tobacco cessation treatment.
Interesting.
Yeah, they had. It's not a large sample size that they used in this study. They had 15 nicotine-dependent smokers, 10 males, around the mean age of 51 years, and many lifetime quit attempts previously. So people who are really trying to quit, and they were smoking a mean of 19 cigarettes a day for a mean of 31 years. 12 of 15 participants showed abstinence at a 6-month follow-up for smoking cessation. So it's a pretty large number a large proportion, 80% of the people. Yeah.
Which is good unless they're schizophrenic.
And it is good except unless they're schizophrenic. The thing to think about here is that with the smoking cessation, normally for behavioral and other pharmacological therapies, the quit rate is typically less than 35% at 6-month follow-up. Mm-hmm. So, why now, I would like to know, does a hallucinogen, which people aren't going to go become addicted to taking psilocybin because that's just weird. Your brain, there's too much weirdness. You're not going to go, yeah, I'm going to do that again tomorrow. Why? Why would a hallucinogenic experience lead you to quit smoking? That's what I wonder.
Well, we've talked about this too, that psilocybin specifically has lasting effects on mindfulness. And perspective, right, and other sorts of things. Used therapeutically, people have had all sorts of interesting, lifelong lasting results. So it's not too too far off the map. The schizophrenia thing, and I'm trying to refine this story. I was reading this earlier today. But they found a link between people who suffer from schizophrenia being able to maintain better cognitive ability and have less sort of episodal hallucinations or less issues, and be able to be more social and more interactive and more stable if they smoke. Nicotine.
Right.
Yeah, it's sort of like the activity in nicotine is it's actually sort of blunted a little bit the connections that can be made in a schizophrenic brain from nicotine that are normally made in a non-schizophrenic brain. However, the effects do seem to be maintaining and allowing better cognitive ability through the illness. So that's sort of interesting. I mean, and there has been, the sort of typical stereotypic, schizophrenic talking about their madcap idea usually comes along with chain smoking. Or at least whenever I'm talking to schizophrenics, they seem to be chain smoking. But I thought that was really interesting. That it could be, they're looking now, they're not going to, of course, nobody wants to prescribe somebody, you know what, you should chain smoke. This is what's going to help you. But having discovered that it may lead to some better treatments.
Yeah. Blair, do you have any more stories?
I have a couple quick things. Nanobots have been developed to test out wine. Nanobots can mimic the reaction that you have in your mouth to a glass of red wine. They can basically mimic the spit hitting the wine in order to measure what they call astringency, which is basically how dry and velvety a red wine is. So they can test that now so that they know more about the wine before they have humans testing it out. Pretty cool. Nanoscience helping us make wine. And then the other awesome piece of science news I have to round out my part of the science news is that deer have been shown to respond to crying of infant mammals of different species. Marmots, seals, domestic cats, bats, humans, and others, all of those infants crying over a speaker attracted the attention of mule deer and white-tailed deer. And they tested the same frequency with non-crying sounds that were made up or were from those species, but they weren't crying. And they've tested other frequencies. They've tested all sorts of things, predatory sounds, and it didn't attract the deer in the same way. So a mammal knows what crying of an infant mammal sounds like, no matter what kind of mammal it is. Pretty hardwired. Yeah.
That's
Good
crazy.
moms are good moms. It's hardwired.
And dads.
Well, these were female deer, Justin.
Well, it works on dads too. I'll just fill that in.
Okay.
That baby crying short circuits my brain. Absolutely. Like it ends whatever thought I was in the middle of, and it absolutely is disruptive. I could drop something. If my baby cries while I'm holding something, I might just drop it. If my baby cries while I'm holding my baby, then they're in trouble. That's on them. But it's something that really is hardwired in to get your attention and make you pay attention to your child.
Researchers have found an object in the universe that has been predicted for about 40 years. They have just discovered it, a Thorn-Zitkow object. Probably not pronouncing that at all. But this TZO was predicted in the 1970s, and what it is a star inside a star. It's like the never-ending jawbreaker of stars. What it is a neutron star, which is an incredibly dense star surrounded by more diffuse cloud of a red supergiant or a red giant. And what they have, the calculations predicted it in the 1970s, but nobody had ever found one before. So some researchers said, why don't we use Hubble data and actually go looking for one and try and match the spectra that we think would be a TZO with what's actually out there. And then they found one. And so what they think happened is that these are the remnants of two galaxies that maybe were close to each other, and they just ran into each other and merged into each other. And the merger creates a system where the neutron star is surrounded by the diffuse envelope of the red supergiant. So it looks mostly like a red supergiant, but the star's core, the neutron, is producing most of the energy from this in the system. And then it's just a matter of mergers of galaxies. And so now they think there might be more out there. There should be more out there, and they're probably going to find them.
Nice.
Yeah.
Awesome.
A star inside of a star.
Inside of a star, inside of a star, inside of a star.
That's crazy, man. That's just crazy.
It's neat. Yeah, it just goes to show how little we actually do know. It's just an odd object in the universe, but it's out there. It was predicted to be there, and it's out there, and who knows what else we will find out in the world.
You guys, maybe our universe is inside of another star.
I think you took the psilocybin pill, didn't you? You did. Did you take the psilocybin pill at the beginning of the show? Is it just now kicking in? Is that what just happened?
I'm sorry.
Oh, for all of you out there. That's right. Different tokes for different folks. Anyone have any more stories? I think we're at the end.
Nope.
Alrighty, then I would like to give some shout-outs to our Patreon sponsors, people who have been supporting us. Thank you so, so much. Kevin Donald, Wesley Ballard, Paul Disney, Sam Winzier, John Ratnaswamy, Rudy Garcia, Aubrey Burgess, Patrick Cohn, Stephen B., Jason Dozier, Matthew Litwin, Eric Knapp, Andreas Akestad, Jason Roberts, Shane and Tara Ginsberg, Brian Condren, Byron Lee, E.O., Bob Calder, Jared Lysette, Ulysses Adkins, Jake Jones, Cassie Lester, Chris Cross, Sarah Chavis, Stephen Soroyech, Layla Amir Sadeghi, Paul Stanton, Marshall Clark, Charlene Davidson Henry, Violated Gorilla, Don Kamarechka, Randy Mazuka, Ed Dyer, Tony Steele, Larry Garcia, Kevin Parachan, Mountain Sloth, Jim Drepo, Darren Lute, Tara Payne, Alex Wilson, Daryl Lambert, John Maloney, Jason Olds, Lord Trentonian Moody, Gordon Grant, James, Paul West, Lauren Lang, Ed Bennett, Thomas Mikinen, Alec Doty, Illumalama, Nick Gradwell, Dan Rambo, Dougal Campbell, TMRO, Miko Pakala, Craig Porter, Adam Mishkan, Jason Martin, Marjorie Scott Lajewski, Philip Shane, Tyler Harrison, Ben Rothig, Columbo Ahmed, and Gary Swinsberg. Thank you so much for your support. We could not do what we do without you. You are so helping us out. And on next week's show, once again, we're going to be broadcasting online live at 8 p.m. Pacific time on twist.org/live. And you can watch live and join our chat room. This is on Thursday next week. But coming in October, I do believe we are going to be attempting Wednesdays, because it turns out that the hosts are having issues with Thursdays. So, October, start of the month, I want you to start setting your calendars, set your alarms, not for Thursdays, but for Wednesdays at 8 p.m. Pacific time.
When's are we doing it?
October is
October 1st.
October 1st will be the first Wednesday date. So the month of October.
When's TWIS on? Is it Wednesday? When's? When's it on? Wednesday?
Wednesdays in the month of October. Justin, what?
Wednesday? Did I say Wednesday enough times?
I think
people
you
heard
did.
Wednesday?
Yes. So next week, Thursday, like normal, but starting starting October 1st, for the month of October at least, if not more, if we find that it works for us better. But for the month of October, starting October 1st, Wednesdays, 8 p.m. Pacific time. But next week, Thursday. So if you're looking for us live, be there Thursday next week. Don't forget to tell a friend about TWIS and to check out our Patreon page if you haven't seen it before, patreon.com/thisweekinscience.
Thank you, everyone, for enjoying the show. TWIS is also available as a podcast. Just Google This Week in Science in your iTunes directory, or if you have a mobile device, you can look up TWIS for Droid in the Android marketplace, or simply TWIS, TWIS, in the Apple marketplace.
For more information on anything you've heard here today, show notes will be available on our website. That's www.twis.org. That's TWIS.org, where you can also make comments and start conversations with the hosts and other listeners.
Or you can contact us directly. Email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or Blair at blairbaz@twist.org. Just be sure to put TWIS, TWIS, somewhere in the subject line, or your email will be spam filtered into oblivion. You can also hit us up on the Twitter, where we are at TWISScience, at Dr. Kiki, at JacksonFly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that came to you into the night, please let us know.
We'll be back here next week, and we hope you'll join us again for some more great science news.
And if you learned anything from today's show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world, so I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice, show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand, and all it'll cost you is a couple of grand. 'Cause this week's science is coming your way, so everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just better understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. Jeopardy. And This Week in Science is coming your way, so everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got a laundry list of items I want to address, from stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say, and if you've learned anything from the words that we've said, then please just remember it's all in your head. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.
gave Kiki fire.
Fire bad.
Fire good! And everyone, it is now the post show. Welcome to the post show from This Week in Science. Yeah, our Wednesday move, I know it's not going to make everybody happy. We can't make everybody happy all of the time. I noticed in the chat room, who was it said? There's something about dog poop and hands. And somebody doesn't have Wednesdays free. Janusku has Wednesdays so full. Replicant X3R0, my every other Wednesdays at the time already taken. And Whiskey Renegade is just upset that Wednesday is harder to spell.
Yeah, it's the stupidest spelling of any
Wednesday.
regular basis. Wednesday. It's
Wednesday.
just dumb. We
Yeah.
should
Okay.
change it.
Yeah. All right. So I need to re-record the show opening. I forgot that I need to do that. Thank you, Identity Force. Let me scroll to the top.
What happened to it?
It did not get recorded. YouTube did not record it. YouTube cut it off.
Boo!
Yeah. So recording in 3, 2, 1. This is TWIS, This Week in Science, episode number 481, recorded on Thursday, September 18th, 2014. Independent variables. I'm Dr. Kiki, and today we're going to fill your head with backyard chickens, a star inside a star, and some psilocybin. But first...
Disclaimer, disclaimer, disclaimer.
All right, we're done now. That's it.
Justin! Take my disclaimer. I liked it.
About to roll
Yay,
it.
Panoramic's baby likes our song.
Yeah.
I like it.
Look at this long, wordy disclaimer on here.
Yeah, I talked a lot. But I was trying to incorporate news, like you said.
So it's a whole story in there.
Mm-hmm.
Yes, so we got it. We're going to be doing Wednesdays in October because I think Blair and I pretty much hashed that out last week, and it's the best idea.
Yeah, well, I'm going to miss 3 Thursdays in October, so...
And I'm going to miss 2 Thursdays. So.
And I'm going to miss 1 Wednesday, so the
I have the potential of
29...
missing every one. To some extent, opening to every one of them.
Yeah.
Oh, Thursdays, you mean. Yeah.
Thursdays. And Wednesday is going to be better for you, right, Justin?
Oh yeah, I can be here. I can be here for the whole thing. The new scheduling, the thing at work, the whole deal. It's ruined me getting here on time to do the show.
So Wednesday the 29th of October, I'm teaching teachers, so I won't be able to be here. I scheduled that before we changed.
It's okay to have one. That's okay. But missing all the way that it was working out with Justin's schedule, my schedule, and your schedule,
there would be no hosts.
There would be no hosts for several shows.
Yeah.
Nope.
Yeah. Actually, on the 29th, I can probably show up late, so I'll try to do that because it goes till 7:30. Yeah, so I'll probably make it around. I could probably make it around the 8:30. So I think my plug worked really well.
You were in here the whole time. Yes.
You guys weren't delayed at all either.
Hardwire for the win.
Yeah.
I am excited about this.
So great.
Can I show off the things?
Yes.
And the stuff now?
Yeah.
Huh?
Boy.
Stuff?
The things and the stuff. This is for us to make better TWIS shorts.
Yay.
We're going to make beautiful video, and it's going to sound really good, too. So everybody, I made a TWIS short about this. So this is in TWIS short number 5. I did a quick briefing. This is it's a DSLR camera, so we can start doing some really neat focus stuff with shorter depth of field and that kind of stuff. Canon EOS Rebel T5i. And people had suggested the T3i, and it's being used by a lot of people who do some really great video, great quality stuff that's up on YouTube. And I went for this also because YouTube is our main video outlet, and so I kind of wanted to do stuff that would look good on YouTube. But this is
on
also
the tube.
Yeah, but it'll records to HD. It can do lower light than the T3i, so get better picture in lower light, and also can record for up to 28 minutes uninterrupted, whereas the T3i was only like 11 or 13 minutes, which is fine for recording a short, but if you just want to keep recording and not cut, if say if we're doing an interview somewhere and it goes a while, you don't want to have it, oh, sorry, I got the camera stopped, so 28 minutes I think will be a good amount of time. So it's this pretty DSLR camera. The battery is finally charged in it. We got an 18 to 55 millimeter lens.
Nice.
Canon lens. So that'll be good. That'll be our workhorse. The kit that I bought, there was a good deal for the kit. It also came with another lens, a 75 to 300 millimeter zoom lens. And I was thinking for this specifically, it might be fun for doing stuff where we're going out and maybe there's animals. Maybe we go to a safari animal park or something.
I know about some of those.
Yeah, we can do zoom stuff with this, which would be neat. Neat effects.
I have connections to a safari park.
That'd be awesome. I also got there a couple of cheapy. There's a wide angle and a telephoto 58 millimeter little cheapy lenses. So they're little crappy lenses, but maybe the wide angle could be interesting for some stuff. The kit came with a little cheapy camera tripod. And then I'm using it actually. I got a little camera topper on an on-camera LED light, so we will have just a light we can take with us, and it's dimmable, so we can turn it down or up depending on the lighting situation. And I also got this beautiful digital field recorder. It is a Tascam DR60D, and this digital field recorder I got because the camera doesn't have really good in-camera recording for audio, so this will help us get really great audio quality for...
That's totally awesome.
Yeah, and this doesn't need to be with the camera. This can do audio recording on its own.
That's great.
It's a field recorder or to camera, and it's got like a slate setting, so you can slate it where you have a slate for the camera, like you clap or whatever. So you can match up the camera to the video to the audio more easily. It's got 2 audio mic inputs, but it has 4 channels, so you can record one a little bit hotter than the other, or 2 channels hotter than the other, so that if 2 of them are too hot and they peak and they have bad audio, the other 2 channels record the same exact thing but a little quieter, so maybe you'll actually get a clean audio recording. There's a lot of stuff with this that I'm excited about learning about how to use it.
That's so great.
And it attaches to the camera so they can sit together.
I need to do a quick experiment because I keep hearing a little bit of audio short static thing coming through. Blair, don't move. Freeze. Don't move. Kiki, jiggle your cables.
Oh, let me move. I put stuff on the cables. It could also be, so I'm jiggling.
Nothing.
I'm jiggling my headphones.
Nothing.
No, I'm jiggling. Is that?
Sounds like that was doing something.
That was my main out cable, though. I shouldn't be jiggling that anyway.
But that sounded like it was tapping on a table.
Yeah.
That's... Okay, Blair. All right,
Yeah.
Kiki, please, don't move.
Yes?
Blair, start jiggling your cables.
Okay, there's that one. There's that one. Those are all of my cables. It's all of them. I don't have many.
So it's not a loose cable connection. So it's something else that's... I better move mine around too. I've only got the one cable now. So it's something else that's hiccuping somewhere. It's not just a loose audio cable of some sort. That's not what's doing it. Well, that doesn't help. Anyway, okay. Oh, here we go. Blair, type something into the chat room. It's not doing anything. Click on something with your mouse. That's not it. Kiki, could you do the same real quick? Just to see if it's something that's hiccuping the audio feed from you that's doing it, or I haven't heard it since we started doing this too. I don't know. No, nothing. Okay, huh?
I'm moving my mouse. Could have been a computer fan that was on or something.
No, it wasn't a fan. It was like it was the kind of like quick static pops that you get like an audio jack being plugged into something that's, you know, how you kind of you twist it and you get that little bit of crackle in out of the speaker. That's what it sounded like. You have a mixing board there, Kiki?
I do.
Can you just wiggle whatever connections are in there, audio jack wise? Just do a little wiggle to it.
I never touch these.
I know. That's nothing. So it's not.
No, there's like one. This one.
Didn't happen. Really weird. So maybe it's maybe it's an internet stream hiccup and not an actual meat space deal.
It's probably connectivity related.
Mm-hmm. Okay, well, that's good to know. Them, not us.
Yeah, WizMic, I decided to get the Tascam because I read that it was better for audio, that not just the Zoom H1 is awesome, but the Zoom SN4 is supposed to be really amazing, like just a great digital recorder. But I like the controls on the Tascam, and it also has quieter preamps so that you can get just really nice, cleaner audio. And I think being able to having the audio readout on the front and then being able to control it all of the levels directly, I think is going to be just really nice. So I'm looking forward to playing with it.
It's going to be great.
Yeah, I did go all the way with my audio. That's right. I was like, let's do it.
So hopefully we'll be able to use that for a short that we're hoping to be filming very soon.
Yeah, next week.
Yeah, I'm gonna get back to you on that.
Okay.
On Monday, hopefully.
Cool.
I'll text you, or I'll text you on Monday with the latest. But I'm hoping it's next week.
Interesting. Okay. Wait. You guys talk for a second.
Okay.
1, 2, 3, 4, 5.
Interesting. Okay. The thing I was hearing is completely on my end.
What is it?
So I go over to my menu, my start menu, and I'm rolling through the options of whatever's there, and as I'm doing that while you're talking, it's interrupting. Somehow that's causing.
It's the scrolly on your mouse.
Now it's not doing it. Hang on. It was doing it, but now it's not.
Oh my gosh, Ed from Connecticut, I love the Neuroscience Prize, Ig Nobel Neuroscience Prize, for trying to understand what happens in the brains of people who see the face of Jesus in a piece of toast. That's awesome.
Now I'm intrigued.
Medicine Prize for treating uncontrollable, quote-unquote, nosebleeds using the method of nasal packing with strips of cured pork. That works?
Yes.
Why pork?
Well, it's salty, right? Pork is very salty.
Mm-hmm.
Maybe there are coagulants in there.
Mm. Seriously, you guys, the digital recorder, I just want to pet it. I know, it's just so funny. I'm like, this little piece of technology, it could have been a nicer, more handheld, easily portable thing, but it's so pretty. And it's going to give us such nice audio. I just want to pet it.
I'm really excited.
Yeah, when we do this short, you better be excited. It's going to make it everything awesome.
I'm just gonna send an email about this right now before I forget. Ask the status. But this organization has been pretty cool about it so far.
About what?
Us filming a short.
Oh, yeah, cool. What flavors did the prehistoric hummingbirds lay eggs? They must have been like those little candy-coated eggs that you get in the Easter basket. Candy-coated almonds. Yes, Identity 4. New audio toys are so fun. It's so exciting. We're going to record audio and it's going to sound yummy. What else do we need to work on? Work out. Wednesdays in October. What are the, is there anything else we needed? Any business?
How's our trip going, Blair?
Nah.
Huh?
Haha.
What do you mean?
I like the NVIDIA post that Genescu put in the chat room. NVIDIA engineers, where did it go?
Dropping things.
The title. How our Maxwell GPUs debunked the Apollo 11 conspiracy theory. Maxwell, the new graphics architecture that NVIDIA is introducing. It is designed to solve some of the most complex lighting and graphics challenges in visual computing. And the demo team rebuilt the scene of the moon landing in Unreal Engine 4, game engine developed by Epic Games, simulating how the sun's rays coming from behind the lander bounced off the moon's surface, Armstrong's suit, to cast light on Aldrin as he stepped off the lander. Yeah. So in rebuilding it, they were able to, let's see, proof that they found. They could reproduce how the light illuminated Aldrin when Armstrong snapped the photo. What else did they find? Photos, details seized on by skeptics. Photos from the landing site don't show any stars, led some to claim the U.S. government faked the landing, blah, blah, blah. But using Maxwell, the demo team was able to find them. And they say the reason the stars aren't visible is the exposures in the camera are set to capture the scene on the moon's surface, but they're there. And the demo team was able to find them by digitally changing the exposure on the shots to reveal them. Woot!
The song's back, Kiki.
Your song is back in your head. Your earworm is back.
Mm-hmm. It was gone the whole show, and right now it's back.
Nice.
I want to know the science behind earworms. Where do they come from? How do I get rid of them?
There is something. Wednesdays with Kiki, Tuesdays with Maury. Saturday and Sunday, the usual alcoholic blackout. CR1, you're funny. That is funny.
It's funny.
There is. We've reported on earworms before. I don't remember.
Okay.
I don't remember all the details about them though.
Should I search TWIS.org for earworm?
Earworm. I don't recall earworm.
We did, I'm sure.
Earworm, I think.
You think it's one word? Earworm? Probably, right?
I don't know.
I'm almost
just
certain.
showing me every episode.
With earworm?
Yeah.
That's not a good search engine.
Is
No, we
earworm kind
couldn't
of
find
like the
any
silence
results.
in Doctor Who? As soon as you speak of it, you forget about it, or as soon as you see it, you forget about
No.
it.
Oh, found one. 31st January 2013.
There we go.
Oh, but there's no there's no show notes.
Of
There's no what?
course. I think you were in Israel.
Oh, yeah. You're totally right. I was still in Israel at the time. Bummer. That's so funny. I found the broadcast and we can't listen to it. Oh, I could listen to it, I guess.
It says repetitive listening could breed earworms. There's an idea that when people listen to music, there's a lot of activity in the motor planning regions. People are often imaginatively participating even while they're sitting still. Imagining yourself dancing? I don't know. So repetitive listening, you know what's going to happen. So if something stimulates something in your head, that it might stimulate your memory of the next note. Song structure might contribute to the earworms. General patterns of characteristics for songs, so they're simple, repetitive, and have some mild incongruity. They tend to have notes with longer durations but smaller pitch intervals. Features that make them easier to sing. So one researcher from Switzerland is saying fundamentally an earworm is your brain singing.
That makes sense. There's an echo.
Is there?
Justin, I think your amazing setup is failing.
Whiskey? No, it's not. My setup is fine.
Echo.
Echo.
Oh. No.
Did it sound like me saying echo, or did it?
Yeah, it did.
That's Whiskey weird. Renegade says, "Are we still having to prove the moon landing was real?" Yes. Whiskey Renegade, people are still trying to prove to me that people that don't believe in the moon landing are real. I don't actually think these people exist. Somebody can prove to me that these people exist, then we can start having a conversation about it. Until that time, I don't even believe these people exist.
Nice.
What are we up against on Wednesdays for scheduling stuff? Wednesdays are just always it doesn't matter. Everything's a busy night. Who are we up against podcast-wise, live broadcasting?
I don't know. He was on Twit.
Yeah, exactly.
Who even cares? I don't care who's up. Well, I don't feel like we have to compete with anybody.
Who's on first? Echo.
Well, there's a minister that has a podcast on Wednesday nights. Bummer. Evening Bible worship. There's like 10 of them. Live church online. Wednesday night Bible study. Wow.
All right.
Coptic Church Network.
Need to create more hangouts. I don't really want to right now. Invite people to next week's This Week in Science TWIS, episode 482. Put that all down there. Hello.
Hello, are you there? What's going on?
Hello. Can anybody hear me?
No.
Is there anybody out there?
Not if you can hear me. Anyone?
So I'm listening to both of you and an old TWIS at the same time, and it's freaking me out. My brain's going to explode.
That's your
So,
echo
how old
there.
is the TWIS? Because if it sounds like this, it's one of the newer twists. If it's me talking, if it's one of the original twists, it's like this because it's like 8 in the morning and I'm on a whole bunch of coffee just so I can be
No,
confident
no,
to
no.
manage to get to the studio on time.
This is January 31st, 2013.
It's
Oh,
an evening,
that's
I'm definitely broadcasting from Israel, so it's 4 in the morning.
which is why there are no show notes.
Yeah.
Okay. At least next week's hangout is now. Next week's hangout is now made. Okay. Now I need to write down the thing.
I sound, oh my God, I'm so hoarse because it's 4 in the morning. It's pretty funny. All right, I'm getting out of here. I'm not going to learn anything about earworms trying to listen to 2 sets of Kiki and Justin at the same time.
No. Good luck with that. Let's see. What should we call this headline? I need a headline for your human origin story.
Three's Company.
Three. There you go.
Three's Company. Okay, great.
I came up with threesome, and then I was like European threesome. And I was trying to be like in Europe,
Whoa.
threesomes are normal, but then that's not a title. That's just trying to make fun of it. How do I? And I know there's a way. But I can't seem to find it.
Femme Fatale bugs. Backyard chickens. Do. Hummer's got sweet.
I'm looking in the chat room. Oh my God.
I need a. Let's see. I need a title for Justin's chimpanzee story. Chimp's just bad.
No. Let's see. Ape in Essence. That's a good one.
Ape in essence?
And Essence.
Ape
I think it's the title of a Aldous Huxley book about
Oh.
the problem of dealing with mankind. I know there's a way to do this. How come I can't find it? Growled Justin. Don't want to. I want to just. It's a simple invert I need to do. I don't want to mess with the color of the thing. I'm trying to fix my photo so I'm pointing the right way. I can't see.
Where?
It's the me pointing picture.
Yeah, but on what? Your Google Plus or something?
I'm gonna try to put it on a Google Plus, but it'd also be nice because if I want to invert the image because I'm pointing the wrong way. I should be pointing at whatever text is on the screen instead of pointing away from it. I pointed the wrong way in the actual picture.
You have 2 overlays on right now.
I do?
Yeah.
Oh, how is that possible?
I don't know.
Yeah, I don't. When I came on, I had to download the Google Toolbox because it was like, you don't have it. I'm like, what are you talking about? I don't have it. Oh, I do. Yeah, and I got the lower third set to off, and then I put it on, and it just puts that there. And the other one I'm stuck with right now. I can't even figure out how to get rid of it. Oh, hang on. There we go. Did that do it?
You still have both.
So yeah, that one's there. I have everything turned to off.
Infant fecal sausages. There's so much wrong with that. Ed. I mean, I know what you're going for there, but
Like, are they infant feces or are they infant sausages? Because
Yeah. There's a lot
they're two
wrong
very
there.
different things.
Very different. Oh my goodness. All right. I need to get going. I have some friends upstairs who are watching out for Kai. Marshall's out of
Oh,
town.
nice.
So I need to go relieve them. They've been very nice to be here this long, but they probably don't want to stay much later because it's getting rather late. They might just be upstairs drunk and disorderly. I don't know. What are you guys doing up there? What's going on? All right, I will be coming back to this stuff. Yeah. Awesome show, you guys.
Yeah.
I think it was a good one. I was glad
I'll see you
Justin
next
could
Wednesday.
come so early on. Thursday next week, Justin.
Whoa. Oh, okay.
Thursday next week. Yes, we have an interview next week. We are going to be speaking with NCSE's Mark McCaffrey. I think I sent you the PDF for the galley of the climate change teaching book that Mark wrote for the NCSE.
Oh, yeah. Okay,
So that's what we're talking about.
great. I will take it.
Teaching climate science. So.
Hey, that's awesome. That's what I do.
Exactly. I thought you might like it.
Yeah.
Yeah.
All right, sweet. Well, that's going to be awesome.
Yeah, so that's next Thursday. And otherwise, I don't know, Blair and I might be shooting a short next week. Might be a lot of fun.
I'll be in touch.
Yeah. All right, everybody. Thank you so much, Justin. I hope you get your picture stuff figured out.
Can't get this decent lower third. It's stuck there. It won't go away. Even if I cancel out of everything.
We're gonna hang up, so it'll be fixed for next week.
All right.
You'll restart before then, I'm sure. All right, everyone. Thank you. Thank you so much for watching, and we look forward to talking with you again next week. Have a great week, everyone. Stopping broadcast.